Hello, my name is Kathleen Tanner, and I decided to do my research project on Huntington's disease. So Huntington's disease is a neurodegenerative disorder that affects individuals from childhood to old age. Most commonly, symptoms are present at mid-age and can be seen through motor impairments or cognitive decline. Among the first to record this disease was George Huntington in 1872. He researched the disease and explained its autosomal dominant pattern of inheritance years before the rediscovery of Mendel's work in 1900. In 1983, the Huntington's gene was first mapped to the human chromosome 4p, but it wasn't until 10 years later that the mutation was successfully identified as a CAG repeat expansion. Translating into a polyglutamine repeat beginning after the first 17 amino acids, and resultingly, this leads to misfolding, aggregation, and toxicity. As recently as 2020, an enzyme associated with the biochemical pathway involving the Huntington gene was identified as TBK1. Tank binding kinase 1, or TBK1, plays a central role in the degradation of the Huntington gene repeat and the clearance of said repeats, as well as the inhibition and blocking of the Huntington gene aggregation. Again, this research I'm reporting on concerning TBK1 is novel to the past few years. In terms of HTT and TBK1, the Huntington gene is found in many tissues throughout the body. However, it is found in the highest concentration in the basal ganglia, a region of the tissue at the base of the brain that controls motor movements. Therefore, HTT and TBK1 can be seen in nerve cells or neurons. In neurons and nerve cells affected by HT, one can observe mast cell death. Figure 1 displays just that. Tissues of unaffected basal ganglia is seen on the left, and HT-affected tissue of the basal ganglia is seen on the right. Notice the difference in the opening sizes of these tissues. For the affected tissue, cell death has eaten away at it, resulting in increased openings of the basal ganglia. In terms of protein pedigrees, I observed both the HTT protein and TBK1 enzyme. Displayed are small segments of the nucleotide and protein sequences for both. Additionally, I included the isoelectric points and molecular weights. And using X-ray crystallography, you can see in both figures 2 and 3 that both secondary structures include alpha helices, and the HTT protein also has coils and loops, while the TBK1 enzyme consists of compacted dimers. In terms of TBK1 and its mechanisms, TBK1 is a kinase enzyme capable of many functions in the cell, making it an ideal enzyme in many biological pathways. With a Km value of 5.9 and Kcat value of 1759, it's comparably more efficient than other related kinase enzymes found in similar systems. Importantly, TBK1 is able to phosphorylate at specific residues significantly faster. TBK1 is known for regulating systems through post-translational modifications, or PTMs. TBK1 also regulates binding partners and associated chemical pathways of the system as described. But we will see shortly exactly how TBK1 regulates the HTT gene. Substrates associated with TBK1 in this system include NT17, which is the 17 N-terminus nucleotide domain of HTT, HTTX1, and long fragment HTT. And as we'll see, TBK1 activation and upregulation aids in blocking aggregation and decreasing mutant HTT in cells. Now begins the specific regulatory properties and mechanisms of TBK1. So TBK1 phosphorylates N-terminus HTT fragments that are regulated by PTMs. And at these fragments, levels of phosphorylation in HD models are reduced to begin with. At these sites, TBK1 stabilizes HTT and blocks further aggregation via site-specific phosphorylation. Next, we can observe autophagy, which is the process by which dysfunctional intracellular structures are degraded. If this process is deregulated, it can lead to Huntington's disease. Importantly, it's known that TBK1 phosphorylates autophagy modifiers and receptors. Resultingly, TBK1 mediates increased autophagy, thus promoting it. In the final steps of TBK1 regulation in mutant HTT, TBK1 clears the aggregation and toxicity from the sites. This begins a process that's promoted by the previous steps. TBK1 signals clearance by activating clearance mechanisms. Some of these include proteasome or autophagy mediated protein clearance. Figure 4 highlights 3 phosphorylation site-specific amino acids within the N17 domain of HTT. These sites being T3, S13, and S16. Importantly, aggregation and decreased levels of phosphorylation have been localized to these first 17 amino acids, which is partly determines why the researchers have chosen these sites. Figure 6 to the right displays a complete diagram of the processes I just described. Phosphorylation, autophagy, clearance of the mutant HTT. In block A, essentially the process of fibrillization of HTT is described, but phosphorylation by TBK1 inhibits this process. In block B, TBK1 site-specific phosphorylation reduces inclusion formation, thus promoting autophagy and autophagy-mediated clearance of mutant HTT. In block C, specific roles of TBK1 at different stages of autophagy are shown, specifically how TBK1 phosphorylates the autophagy adapters, so that's P62 and OPTN, that bind the substrates or damage mitochondria. Next, if you look in block D, it simply indicates that one thing to note is that TBK1 doesn't phosphorylate pre-formed mutant HTT inclusions or fibrils or promote their clearance or dissociation in vitro. Additionally, to observe overall results of TBK1's role in the system, you can see in figure 5, the cell death, represented as a percent. So cell death is significantly less at HTT sites with active TBK1 versus inactive TBK1 HTT sites. Those specific sites, as indicated, are HTT-X1, 16q, and HTT-X1, 72q. And today, management of Huntington's disease remains challenging, and in most cases, utilizes off-label treatments. The only drugs that are US Food and Drug Administration approved for Huntington's disease right now are tetrabenazine and deutetrabenazine, which are used to treat chorea. So chorea is a neurological disorder and influences the involuntary movement and jerking of the body in the shoulders, hips, and face. So while there is a lack of significant treatment options available for Huntington's disease, there is, however, hope surrounding gene modulating therapies due to research just like the research I described, done on TBK1. Additionally, it is not well known that TBK1 has recently been involved in COVID-19 research due to its role in immune response and antiviral activity. As recently as February of 2022, Gontier and others of Research Square published novel data suggesting that inhibition of TBK1 decreases the severity of COVID-19 symptoms. I was pretty surprised to read that because in terms of immune response and antiviral activity, TBK1 activates other regulatory factors associated with immune response. So I would think that TBK1 activity would be beneficial, but this research suggests just the opposite. In fact, it's possibly an issue over excessive activation leading to negative effects in these pathways. So with all that being said, my sources are listed to the bottom right, and thank you for listening.